It's my pleasure to introduce Brooke Trimmer, BS. She is a biochemistry trained scientist with 4 years of experience in the biotechnology and pharmaceutical industry, specializing in analytical characterization of both biologics as well as small molecule parenterals. Her work focuses on applying advanced flow imaging and particle characterization techniques in order to support product quality, comparability assessments, and method robustness. She's contributed to cross-functional development programs which span early research through late-stage development, partnering with formulation, quality control, as well as device teams to generate high-quality analytical insights. Brooke is especially interested in bridging laboratory science with real-world applications and in developing practical, data-driven approaches that make complex analytical methods more accessible to broader development teams. So with that, Brooke, the floor is yours.
Great. Thank you so much, Tyler. Like Tyler said, my name is Brooke Trimmer. I am here at Gilead Sciences, and I work in the particle characterization team. And I do a lot of flow imaging, which is why I'm here today to tell you about advancing parenteral development through flow imaging microscopy. So for today, I wanted it to be really practical. Here, just trying to get my slides to advance. It's just lagging. Um, while that tries to load, there we go. Oh, there we go. Um, so today I really wanted to focus on a very practical webinar. So what we're going to first start with is just an overview of what flow imaging microscopy is, where does it fit in, and then we're going to talk about generating positive controls, doing some sample analysis, what to do if you have an investigation, and then just kind of wrap up and go to our Q&A. So to start, I love this. This is from Jamie Moore. She has this is kind of like her iconic slide here with keep calm, particles happen, right? If you're here, you probably have to do some sort of work with particles and particulate matter analysis. So, flow imaging has been a really wonderful tool in helping us to do this sort of characterization. And so to kind of start where I think a lot of people are, flow imaging microscopy kind of fits in with our 788, USP1 788, method 1 USP 788 tests and method 2 788 tests. So that's things like light obscuration, which is highly quantitative, right? So we do that with the HIAC, and it's a bit of a black box method, right? So we put our sample in, we get a number out, but we don't really know what those numbers mean. And then we have method 2, which kind of goes hand in hand with light obscuration. We'll call it semi-quantitative. It's kind of quantitative, but it's not quite as robust quantitatively. And it has a lot more qualitative data that you get, right? You get actual images of your particles. And so there's a little bit of a gap between these two methods that I think dynamic flow imaging fits really nicely into. Even though it's not yet a formal USP method, it is discussed extensively in the informational chapters in 1788 and 1788.3. And you can use it orthogonally to, but not a replacement for, but you can use it orthogonally to method 1 and method 2. So it's a really nice kind of bridge between these two techniques that can help kind of support your data. And then odds are if you have microscopes or a HIAC in your lab, you're probably doing some particle characterization work. And so I just wanted to kind of take a step back and kind of look at our whole toolkit, right? And how flow imaging kind of fits in as one of our tools in subvisible characterization, right? So there's a lot of different tools and techniques that we use. Orthogonal techniques are super important in this field, right? To help verify what we're characterizing. And so this is like our Swiss Army knife, right? This is just one of our tools that we can use in lab to help characterize.